much for inviting me to be here. And, that was a very hard act to follow. But I'm going to spend the next 20 minutes giving you kind of an overview of some innovations in genetics and genomics. So we've come a long way since 1859, when Charles Darwin first published his book on the origin of the species based on natural selection. Soon after that, Gregor Mendel, who was an Austrian monk who did very careful crossbreeding experiments on peas, told us that heredity was transmitted in units. And fast forward to 1953, when Crick and Watson described the structure of DNA as a double helix. We're now at the 70th anniversary of that discovery, and things have gone very fast since then. In the 1950s to 1960s, Kornberg isolated DNA polymerase, which would later be used for DNA sequencing. And then in 1966, Nuremberg, Corena, and Hawley cracked the genetic code. And in 1968, the first restriction enzyme was described, which has become a tool for mapping genomes. And then in 1975, a huge breakthrough with Sanger, Maxim, and Gilbert developing rapid DNA sequencing. This was followed by discovery of polymerase chain reaction, or PCR, which we use today to amplify DNA. And all these led to 1977, the first genome being sequenced, which was in a bacteriophage. And then 10 years later, in 1987, the first generation human genetic map. A huge development in genetics and genomics was the launching of the Human Genome Project in 1990. This was a very ambitious 15-year project to sequence the human genome, all 3.2 billion pairs of the human genome. And that led to, in 1992, the second generation map of the human genome. And then by 11 years later, publication of the first draft of the human genome. And the project was actually finished in 2003, just 13 years later, 2 years short of its target ahead of time, which is pretty uncommon in science. In any case, that there was a lot of technology and tools that were developed through the Human Genome Project, including development of next generation sequencing, which allowed us to then proceed to more in a faster way sequence whole genomes. And so that there was the launch of the 1,000 Genomes Project in 2008, which aimed to sequence 1,000, and they actually accomplished sequencing over 2,000 genomes and across different ethnicities, and also with the goal of getting a whole genome sequence under $1,000, which we have achieved. So, and this led to deeper sequencing, such that the completion of the human genome sequence was just published last year in 2022. So the Human Genome Project has had huge impact. First of all, it was a model for cooperative, collaborative, large-scale science. It involved 20 academic centers across 5 countries. It led to things like the Encyclopedia of DNA Elements, or ENCODE, which was done by a public research consortium aimed at identifying all the functional elements in the human and mouse genomes. Also, the International HapMap project to study human DNA sequence variation. It led to the era of genome-wide association studies, or GWAS. And again, as I mentioned, a lot of tools and technologies, including next-generation sequencing and being able to sequence genomes individually. So the era of genome-wide association studies, it has been pretty transformative. We can now assay hundreds of thousands and millions of SNPs, or variants, across the DNA. And these variants are called single nucleotide polymorphisms, or SNPs. So you can do very large case control studies where you take individuals with a disease, like in the left column, and individuals without a disease, your controls. And then you see which SNPs or variants are associated with your disease. So if you see here in SNP1 and SNP2, there's pretty equal distribution of the SNPs between the cases and the controls. But if you look at SNP3, that has the people with the disease have a lot more of those variants than the controls. So you can, actually this is a way where you go from the bedside to the bench, because here you hear the patients actually telling you what's associated with their disease. This is a GWAS for Crohn's disease, and often these GWASs are displayed as what's called a Manhattan plot. So on the x-axis is each chromosome, and each of the dots is a SNP, or a single nucleotide polymorphism, a variant across the DNA. And on the y-axis is the significance level, or p-value, a negative log of the p-value. And you see that there are several areas on this that rise above the noise. Now, I work on atrial fibrillation. So in the early 2000s, we started gathering a lone atrial fibrillation gene bank with atrial fibrillation in the absence of structural heart disease. And we found that, that there is this very strong hit on chromosome 4q25. Decode Genetics actually published this. We contributed our data to large consortia, and by 2018, with over 80,000 cases of atrial fibrillation, over 100 AFib GWAS loci were discovered. And this year, we should be publishing another one that will have over 200 AF GWAS loci. But through this all, this locus on chromosome 4 has been the strongest. And you can see on the bottom that it's over, the p-value is like less than 10 to minus 500. So this top locus is near a gene called PIDX2, which happens to be involved with formation of the pulmonary veins. And as you can see here, that's what we're ablating for atrial fibrillation. We're isolating the pulmonary veins. Now, we wouldn't have discovered this because we thought atrial fibrillation was an electrical disease. So all of our drugs have been pretty much focused on ion channels. And so here is an example where the patients actually tell us that, hey, there's more involved than just ion channels in atrial fibrillation. So as of 2022, the GWAS catalog has over 45,000 published GWASs across over 5,000 traits. And this is just an amazing thing, from prostate cancer to hypertension, height, etc. Next generation sequencing has also been very transformative. This is massively parallel sequencing technology. It's revolutionized our biological sciences. We can now rapidly sequence whole genomes, deeply sequence target regions. We can do whole genome RNA sequencing, so genes are transcribed to RNA, and we can then see how different diseases affect gene expression and which genes or SNPs may be causative. We can study epigenetics, which includes genome-wide DNA methylation and DNA protein interactions. We can sequence cancer samples. We can sequence the human microbiome, and we can identify novel pathogens, which is really important nowadays. The advancements in cancer genomics has been amazing. There's a cancer genome atlas from the International Cancer Genome Consortium. The first cancer genome was in acute myeloid leukemia that was sequenced in 2008. And we use next generation sequencing and targeted next generation sequence panels for tumor profiling. But it's also been used to identify therapeutic targets. For example, breast cancer, HER2 amplified breast cancers can be treated with trastuzumab and BCR-ABL fusion positivity and CML imatinib. Non-small cell lung cancer can be treated with mutations in EGFR, ALK, and ROS1. Melanoma patients who have mutations in BRAF can be treated with inhibitors of the MAP kinase signaling pathway. And metastatic colon cancer, there's some EGFR-directed therapies in patients without oncogenic mutations in KRAS or NRAS. And this list is expanding. I'm not a cancer person, but it's just amazing what has been developing. And transcriptomics, I mentioned mRNA, and we follow this paradigm, genes to function. DNA is transcribed to mRNA, which is translated to protein and function. And these associations can help us identify the genes and the gene variants that are causative. Epigenomics is the systematic analysis of gene expression that's not attributable to mutational changes in the DNA genome. And we can look at cell-specific epigenetic marks that are reversible, and they affect gene expression, but they don't change DNA sequence, and they can be heritable. So DNA methylation in the yellow generally results in gene silencing, Histone modification can lead to gene activation, silencing, or insulation between inactive and active gene regions. They can change the nucleosome density to being open or more compact, making them more or less available for transcription. And similarly with chromatin structure, the higher order chromatin structure can compact the nucleosome often near the nuclear membrane, or create large loops that compartmentalize the genes and bring together enhancer promoter interactions. These epigenetic changes can be affected by environmental factors like smoking, diet, toxins, stress, exercise, aging, or mutations in the epigenetic modifier genes. And we can detect them sometimes in blood or tissue. And in cancer, that's been the paradigm of common epigenetic disease. So we can look at biomarkers for epigenetics, and also there's work looking at drugs that target. Now, these advances in multi-omics and next-generation sequencing has really impacted immuno-oncology using exome-wide targeted DNA, tumor versus non-tumor, whole genome, and RNA sequencing, and even single-cell RNA sequencing, and it's all beginning to impact clinical management. For example, with immune checkpoint inhibitors that can target somatic mutations, that is, it's not in the gene line, and cancers that have high mutation burdens have responded well to immunotherapy. Anti-cancer agent, for example, pembrolizumab, is based on a cancer-specific genetic profile rather than the tumor site, lung versus liver, etc. So next generation sequencing-based discovery can thus compare these tumor versus non-tumor DNA to identify somatic variants. You can design anti-tumor vaccines targeting peptides, RNA, or DNA, or dendritic cell vaccines. So these developments in multi-omics has also been paralleled, as we heard, by advances in data sciences. We need that because there's a lot of data being generated. So we need the bioinformatics, systems biology methods to be able to layer on in our analyses, not only genomics, but epigenomics, transcriptomics, proteomics, and metabolomics. So we, as I said, study atrial fibrillation, and we've just published this trying to go from genes to drugs. We've used some network systems biology and AI methods to try to identify reproducible drugs for AFib. So we've taken the human interactome, the protein-protein interactome, layered on genomics, genetics, and RNA sequencing in tissues, and about to layer in metabolomics and proteomics. We can identify an AF gene module. And then we can use drug target databases and use a closest distance approach to see which drugs target the closest genes to the AF disease module. And here we've identified a bunch of drug targets, and this shows the 55 top drug candidates nominated by this that we can now test for atrial fibrillation. So genome editing is also a very exciting development, and it's used bacterial nucleases and zinc finger nucleases, transcription activator-like effector nucleases, and you've probably heard about CRISPR-Cas9, stands for clustered regularly interspaced short palindromic repeat associated 9 nuclease. And these are like genetic scissors. The aim is to make double-stranded breaks in the DNA, and you delete out your sequence of interest and insert in what you want to replace for that. And CRISPR-Cas9 uses RNA guide sequence to do that. And in 2020, the Nobel Prize in Chemistry was awarded to Emmanuel Charpentier and Jennifer Doudna. So the applications have been widespread already. We've used it for disease modeling. We can make genetically engineered cells and animal models to study molecular mechanisms that can then be targeted for therapies. And in cancer research, with generation of leukemia, organoid tumor models to study functional genomics. And cardiovascular disease, we can create research models. And heart disease, it's especially hard because you can't take biopsies of tissues, but you can actually pair this with stem cell technology and CRISPR edit in your mutations of interest. You can also ablate MHC class 2 for allograft engineering, and it has a potential for gene therapies in inherited diseases. It's been used in metabolic diseases to establish model organisms in obesity, diabetes, and lipids. And there's more, as I mentioned, in inducible pluripotent stem cells. which can be called iPS cells. And in 2012, the Nobel Prize in Physiology or Medicine was awarded to Sir John Gurdon and Shinya Yamanaka for discovering that you can reprogram somatic cells, mature cells, to become pluripotent stem cells again. So they took skin fibroblasts, or Dr. Yamanaka took skin fibroblasts and incubated them with 4 transcription factors that we call Yamanaka factors, and that could turn them into stem cells. So you can also do this not just with fibroblasts, but with a regular blood draw, with white blood cells. You can take white blood cells and reprogram them into stem cells. The applications of stem cells are wide. They range from regenerative medicine to using these as disease models in drug screening, because you can differentiate these cells into various cells like nerve cells, cardiac cells, liver cells, beta cells, lipid cells, adipocytes. And this is just really on our way toward real precision, patient and mutation-specific therapies. So as I said, I work in atrial fibrillation, and we can use this to make actually beating heart cells. And you could hopefully see that you could generate sheets of cardiomyocytes that actually beat, and they actually beat. You could actually sort of see the ripples going across our cell dish here. They contract so much that they kind of pull off the media, which is a problem for us actually, and then we can actually form them into engineered heart tissues and exercise them and study their electrical properties. So you could actually take these, normal human iPS cells and use CRISPR-Cas9 to edit in your mutation of interest, and then you have the normal wild type in a nice pair to study. And you could also take specific mutations from patients and edit back in the wild type. In this way, you have these sets of these pairs of cells with and without the mutation. It's a very powerful model that you can study electrically or omically, and also do drug targeting and drug toxicity testing. There have been also clinical trials using gene editing in the treatment of human diseases. Sickle cell disease is a big model for this, with different blood disorders, cancers, CAR T cells, checkpoint inhibition, inherited eye disease, diabetes. You can take type 1 diabetes, CRISPR-edited pancreatic cells that are derived from stem cells and put them back in a patient. Infectious disease, inflammatory disease, and protein folding disorders like amyloidosis. CAR T cell therapy in immuno-oncology, again, is, it's been a model of this. You can take genetically engineered chimeric antigen receptor, or CAR T cells, to target a tumor-associated antigen. So you take a patient's own white blood cells, and then you isolate the T cells and you re-engineer it with tumor antigen-specific receptors and co-stimulating molecules. You amplify them and you infuse them back to attack their tumor cells. Now, this has also been a very exciting development with in vivo CRISPR editing used for amyloidosis. There's a report just a little over a year ago with 6 patients that had hereditary amyloidosis and polyneuropathy. And they had the CRISPR RNA guide sequence for the TTR mutation. They put it in a nanoparticle that was targeted to the liver through the LDL receptor. And it actually decreased the amount of the mutation that was transcribed. It was just really amazing. And they've also used it and reported that in the news, at least, that they can use it for cardiomyopathy. I never actually thought I was going to see in vivo CRISPR editing in my lifetime, but here it is. And we've seen advances in genetics and genomics applied to SARS-CoV-2 during the COVID-19 pandemic. As you know, in November 2019, there was the emergence of COVID-19 from Wuhan, China. And then just a month after we were learning about it, the whole genome was sequenced and shared. And ACE2 was confirmed as the host cell receptor for SARS-CoV-2 spike protein. And the monoclonal antibody was engineered and designed. And the first clinical trial reported on October 28th, 2020. And by November 9th, 2020, FDA issued the first emergency use authorization for the first monoclonal antibody. And of course, we have mRNA and DNA vaccines. And the first vaccine that we got an EUA for was just in December 2020. So less than a year after this came out, we had our vaccine. Normally, this takes 2 to 5 years at least to develop. So in summary, there have been tremendous strides in genetics and genomics that have been made over the past 70 years since Watson and Crick reported the molecular structure of DNA. And our rapid advances in sequencing technologies, multi-omics, big data analyses, iPS cell technologies, and genetic engineering are really revolutionizing the biological sciences. And these advances are already coming to our bedside to help mitigate or even cure selected diseases. So thank you very much for your attention. I can take any questions.